All right, so this is lesson 10 in unit 2. Okay, this is our last lesson in the organic chemistry unit. It's an extension of the previous lesson. We're still on polymers, except this time we're going to talk about condensation polymers. Okay, we're going to talk about synthetic condensation polymers, and then we'll talk about natural polymers. And within natural polymers, we're going to talk about the different biological molecules, polysaccharides, polypeptides, and nucleic acids. So let's start with condensation polymers. So we learned about addition polymers. It's simply double bonds broken, and they all add up to one big thing. A condensation polymer is still a long molecule, except it is formed via condensation reactions between two different functional groups. Okay, and we learned condensation in a previous lesson. It is simply the removal of water from the two molecules, and then you will then link them together. So the diagrams on the bottom of the slide, the condensation between butanoic acid and methanol, as well as butanoic acid and methylamine, that is the exact same picture that I showed you earlier. The esterification reaction and the reaction that produces an amide. These are condensation reactions, and they form polymers if you're able to somehow put them into the same chain. So just a little review. To make an ester, you need an acid and an alcohol. And if you remove water from those, you will make an ester linkage. To make an amide, you need an acid and an amine, and if you remove water from that, you have an amide linkage, okay? Now, so let's actually look at these two types of condensation polymers. So the first one is ester. So polyester is a polymer formed from the condensation of monomers that are linked together via ester linkages. The word polyester is actually a common English word. Okay, you don't need to have a working knowledge of chemistry to know what polyester is. It's a type of fabric in different clothes. So we're going to learn the chemistry behind polyester. So a polyester is just a polymer of esters. You would have a molecule that has both a carboxyl group and an alcohol, and that somehow linked with itself over and over and over again via ester linkages. Okay, kind of like the picture shows right there, the carboxyl and the hydroxyl, you lose water and you have an ester. The common polyester that we have in fabric is Dacron, and Dacron is basically a polymer of benzene 1,4-dicarboxylic acid with ethane 1,2-diol. Now, those names are actual IUPAC names. Benzene 1,4-dicarboxylic acid is just a benzene with 2 carboxylic acids, one on each side. So you have 2 acids. The diol is an ethane, 2 carbons, with an alcohol on each carbon. And as you can see, this can repeat. You can have a benzene and an ethane, followed by another benzene, another ethane, another benzene, another ethane, which eventually will get you a polymer that's what we call polyester. So the monomer of Dacron is on the bottom. You have a benzene with carbon double bond and another carbon double bond oxygen, followed by an oxygen and an ethane molecule. So that is actually an ester linkage. So this is the structure of what we call polyester, and we use that in t-shirts, hats, sometimes pants, socks. So that's a common thing. You can just pick up polyester material at Walmart. Polyamide is similar to polyester. The only difference is instead of an oxygen, you have a nitrogen. All right, so polyamide is just a polymer of amide linkages between the monomers. They are more stable and durable than polyester. Okay, as a result, we can use polyamide in a lot of other applications other than clothing. So for example, Nylon 6,6 is a polyamide, and I'm sure you've heard of something called nylon before. You can use that in clothing, but a better application would be to use it in say, ropes and something else we'll talk about later. So you would have nylon 6,6 in the picture. You actually have hexane with not carboxylic acid, but it's something similar. Instead of C double bond O with an OH, you have a C double bond O with a chloride. This is polymerized with hexane 1,6-diamine, so a hexane molecule with two amines on the ends. Instead of removing water, you remove HCl. All right, so you will still have an amide linkage. The carbon double bond O, circled in green, will bond with the nitrogen, also circled in green, and you form an amide linkage. So this on the bottom will be the monomer for nylon 6,6. Okay, so that's what we mean by nylon, the fabric, that structure right there. I alluded to this on the previous slide. Another interesting application of amide is Kevlar. Kevlar is a material we use in bulletproof vests and as well as aircrafts because of its strength. Why does this material have so much strength such that even bullets can't penetrate? Okay, heat will not disrupt it. What gives? Well, this is the structure of Kevlar on the right. It is simply chains of polyamide. The special part is the chains are kind of connected with each other, not by London forces, but with hydrogen bonding because of the presence of NH bonds and C double bond O. They're able to make hydrogen bonds with each other, and hydrogen bond is the strongest intermolecular force. So you will need a lot of force to break that bond. So a speeding bullet, the kinetic energy of the bullet is not enough to disrupt these bonding. Okay, and you need a lot of heat in order to break that. So this is highly resistant to heat as well. So that is a pretty interesting application of polyamide. All right, so here comes example one. Example one refers to another type of nylon. So draw the structural diagram and write the names of the monomer used to synthesize nylon 6,8. Okay, so what is nylon 6,8? Well, there's a clue. Nylon 6,8 is made up of a 6-carbon diamine and an 8-carbon dicarboxylic acid. All right, so I'm going to give you some time to work on this one. I'll take it up in 2 to 3 minutes. All right, so you would have something like this. To draw this nylon monomer, you have to draw its components first. And the clue is a 6-carbon diamine. What does that look like? Well, it looks like this. 6 carbon, so hexane. Diamine, it has 2 amines on it. So the natural thing is to draw the hexane 1,6-diamine molecule. Make sure that the 2 amines are on terminal carbons, okay, because you need to make a chain. If it's on a middle carbon, you're not making a chain. So at 8-carbon dicarboxylic acid, well, 8 carbon means octane. Dicarboxylic acid, meaning that you have 2 carboxylic acid, 1 on each end. Right, so now you have an amine and an acid. Now they're able to make an amide. You will undergo condensation, remove the OH from the acid, remove a hydrogen from the amine, and you can connect the carbon to the nitrogen. So this will generate a peptide bond. Okay, just like in biology, this is called a peptide bond, also amide linkage, exact same thing. And if you were to just repeat this, this will become a polymer of nylon 6,8. Okay, so this subunit right here, this whole thing will be the monomer. Okay, does that make sense? So this is quite a difficult question. You have to basically figure out how to draw the ingredients that makes up the monomer So once you get that, you can tell me what the monomer is. So do we have any questions? All right. All right, so let me just explain a little bit about unwanted plastic. So there's a lot of plastic waste right now in this world. Plastic was a great invention, okay? It made our lives so much more easier and convenient. Plastics are made up of polyethene, also known as polyethylene, as well as polystyrene or styrofoam. The reason that plastic is so good is its high stability, okay? It will not easily react with a lot of common substances, hence it is a good material to use for containers. And they don't really break down over time, and they're quite cheap to produce too. They're cheap, they're light, they don't break easily if they fall, and they're very stable. So plastic has become a staple of our lives. Problem is, the greatest strength of plastic is also its greatest problem. Because it is so stable, once we're done with the plastic and we chuck it out, it's going to stay there in the landfill for thousands of years because plastics don't really degrade. All right, we like it because it doesn't degrade, but once we're done with it, we want it to degrade, and it still wouldn't. And a lot of plastic that we have is just a one-time use thing, like a plastic wrap. If you go to the supermarket and you want to pick up some fruits, you can likely see the fruits wrapped up in some kind of plastic. So you buy that home, you take out the fruit, you eat the fruit, you chuck the plastic. Okay, that's it. That's gone. So there's a lot of plastic waste in this world. We generated so much plastic. In fact, if you look at that globe on this page, this is called the Great Pacific Garbage Patch. And there's a lot of plastic there. Yeah, it's that big. And a lot of water has been polluted with plastic. So a lot of aquatic creatures like turtles, whales, other fish, they will swallow by accident a lot of plastic. Sometimes on purpose, because animals, they can't really tell. I've heard a news report about a whale that got washed ashore and died. So people did an autopsy, cut the whale open and opened the stomach, and discovered tons of plastic in the stomach of the whale. So how a whale eats is that the whale opens its mouth and just starts swimming. Things would just get sucked in, including the plastic. The fish will get digested, the plastic won't. So the whales will just accumulate plastic inside of his stomach and eventually the bloodstream. Okay, many birds died because they ingested too much plastic. And because they cannot digest the plastic, it is stuck in their stomach. And that takes up space. So the bird is bloated in plastic, very hungry because of a lack of nutrient, but the bird can't eat because the stomach is full of plastic. So you starve to death. So there's a lot of disruption and death in the ecosystem because of unwanted plastic. So the Canadian government decided to slowly weed out the use of disposable plastic. And I am in favor of this resolution. I think we can live without one-time use plastic. It's going to make our lives a little bit inconvenient, but there are alternatives, okay? We can live without plastic. So what are some alternatives? Well, there's biodegradable plastic. Plastic, the conventional version, is basically a giant alkane molecule. It is made up of addition polymers, okay, polyethene. But if you can manage to make plastic with condensation polymers, like using ester and amide linkages, well, because they have now functional groups, that becomes a lot more reactive. Okay, and an alkane is just carbon-hydrogen bond, carbon-carbon bond. That is not reactive. You're not going to break that. But microorganisms like bacteria, they are able to break ester and amide linkages. So if you have a biodegradable plastic, you can chuck that out. Microorganism will slowly degrade that, and eventually it will just be assimilated by the environment. The contents will be recycled. Okay, so it has a less harmful effect on the ecosystem. Of course, the best way is just to simply use less. Now, if you have less consumption, there will be less production. And if you have less production, well, you have less garbage. So you just look at what happened to the oil prices during the pandemic. Right? People started driving less because people stay at home now. So the gas prices plummet. Okay. A lot more oil is being produced than being consumed, so the price got lowered. Eventually, they adjusted the supply, so the price started to go back up again. So you can see the effect of supply and demand. If you demand less plastic, there will be less plastic produced, hence reducing our garbage output. If the garbage that we have already put into the environment, yeah, that's really hard to clean up, so we got to make sure that we don't continue to do so. All right, so let's talk about natural polymers now. So we talked so far only about synthetic polymers. That means we humans manufactured that. Okay, they're not found in nature, but there are polymers in nature. There are polymers in your body. Okay, so there are 4 major biological macromolecules in living organisms. And you learned this in grade 12 biology if you took that course. That's the first unit in grade 12 biochemistry. We're basically going to summarize biochemistry in a few slides. The 4 major macromolecules include carbohydrate, lipid, protein, and nucleic acid. Lipid is kind of different because lipid is not really a polymer. Okay, lipid is a triglyceride with a glycerol attached to 3 fatty acid chains. They're bonded together using ester linkages, but that's it. That's a lipid molecule right there. Okay, it's not really a polymer. We're not talking about the lipid tail, the repeating carbon. No. So we're not including lipid in our lesson here. So we're going to be focused on carbohydrates, which are made up of polysaccharides, proteins, which are polypeptides, as well as nucleic acid, which are polymers of nucleotides. All right, so the first one is carbohydrates. A carbohydrate, as the name suggests, carbon, hydrate meaning water, so it's just carbon with water. Right, so the formula is CnH2ON. The primary carbohydrates that we're going to look at are glucose and polymers of glucose, so starch and cellulose. Starch is the storage molecule for energy for plants. Okay, like potatoes has a lot of starch. We eat potatoes, hence we derive glucose from the starch. Cellulose are components of cell walls. They're extremely strong, and that's what makes cell wall strong because of the structure of cellulose, which is a polymer of glucose. A carbohydrate is made up of polysaccharides, which are made up of monosaccharides. Okay, mono means one, saccharide in Greek means sugar, so literally this says one sugar. And the most common one is glucose. Glucose is an aldehyde or a ketone with 5 or 6 carbons. Okay, not necessarily glucose, but a monosaccharide. But glucose is a 6-carbon molecule. I'm sure that you've seen the ring configuration of glucose on the left side in biology class. The middle is the chair configuration of glucose. Okay, it kind of pays more respect to the Vessel geometry. I don't know if you've seen the chain configuration. If you stretch out glucose, that 6 carbon, and you break up the ring, you put it in a chain, well, you have an aldehyde group on the top and a bunch of hydroxyl alcohol groups. Okay, so these are all different representations of glucose. To go from the chain to the ring, all you have to do is loop it around, do a condensation reaction between carbon 1 and carbon 5, and you would have the chair again. So a polysaccharide is a polymer of monosaccharides, which are formed, we're using glucose in this example, by long chains of glucose molecules. Okay, and there are 3 major types of glucose polymers. First one is starch. And like I said before, starch is the energy storage molecule in plants. And they're held together by glycosidic bonds between 2 glucose molecules. It's like the glucose all just hold hands with each other. Okay. However, this can be branched. Right? It doesn't have to be just a straight chain. Starch actually has a lot of branches just coming out from the side here. So it's like a web of glucose. That will be a polysaccharide for plants. The animal version of starch, the thing you have in your liver, is called glycogen. It is exactly the same as starch in terms of composition, chains of glucose molecules. Glucose holding hands with each other to make a polysaccharide. It has more branching than starch, but it serves the same purpose. Right? So if you eat a lot of food, you might not metabolize all of the sugar that you ate, so that will be stored in your liver as glycogen for later. So if you get stuck on an island with no food, you're not going to die immediately. You will start to exhaust your stock of glycogen in your liver. So you will still go on living with that glycogen you stored up. If you exhaust that, you start to burn the lipids. After you exhaust that, you start to burn the proteins and the muscles, which you really don't want to do, and then you starve to death. Okay, so it's a long process. So glycogen is the energy storage molecule inside of animals. Cellulose is a little bit different. It's still all glucose, but this time there's no branching. Okay, all the glucose hold hands with each other, and they make a long chain. That's it. Because it doesn't have any branches, it is able to neatly stack on top of each other, and those stacks are held together via hydrogen bonds because glucose has a lot of OH groups. And the strong hydrogen bonds, as well as the glycosidic bonds between the glucose molecules, make cellulose very, very tough. Okay, hence cell walls. Not a lot of things can dissolve cellulose. Humans lack the enzyme to break down cellulose, which is why eating grass is not a good idea. You're not going to get full on grass because we can't metabolize grass. So example 2 makes you draw a polymer of glucose. So starch is a polymer found in the condensation reaction in which glucose molecules polymerize via condensation. Draw a starch with 3 glucose monomers. So I have provided the structure of glucose. Your job is to put them in a chain, hook them together to make starch. All right, so I'll give you 1 to 2 minutes for this. You have to think about what you have to do. Okay, a clue is this is a condensation reaction. You have to remove water. So I'll take this up in around 2 minutes. All right, so to draw starch, how to connect the glucose together. Well, it's a condensation reaction, so I'm highlighting the hydrogen and the hydroxyl from the 2 carbons that are on the side. One of them will lose the hydroxyl, the other one will lose the hydrogen. Same thing between these 2 glucose. One will lose the hydroxyl, the other will lose the hydrogen. Once you do, you make 2 water molecules, and you can just link up the glucose together, and they're held together by 1 oxygen atom. It's like they're holding hands. So this will be the drawing of starch. Okay, does that make sense? You do not have to go into as much details as you did for biology. We're simply looking at the chemistry basis for this. All right, so for the next type of macromolecule is proteins. And proteins are polymers of amino acids. Hold up. And amino acids are small molecules made up of carbon, oxygen, nitrogen, sometimes sulfur. So there are different kinds of amino acids. So the picture on the bottom shows you the general form. There's an alpha carbon in the middle. And the alpha carbon is making 4 bonds. One of the bonds is with a hydrogen on the bottom. Another bond is with an amine group, NH2, on the left. Another is with the carboxylic acid, a COOH, on the right. And finally, the last bond is with an R group. What is the R group? Well, the R group can be a lot of different things. Actually, it could be 20 different things. There are 20 different amino acids because they all have a different R side chain, which gives them different properties. If you take these amino acids and you just link them up together in amide linkages, you will have a protein. Right, so here is a diagram of all 20 amino acids. On the first row in blue, you would have the charged R group. So these amino acids have different properties because of the different structure in their R groups. For the first one, they're all charged. You either have positive or negative charge. And if you're charged, then you will be attracted by water, okay? Because water is highly polar. You will dissolve in water, so hence these are hydrophilic amino acids. The row in the middle, they're polar R groups. All the R groups contain oxygen or nitrogen or both, so they are polar side chains, also being hydrophilic, because in grade 11 you learned that like dissolves like, polar substances will dissolve polar substances, and water is polar. On the bottom row, you have the non-polar hydrophobic side chains. These are all carbon-hydrogen bonds. All right, and we learned in the first unit how this is non-polar because of symmetry and a small difference in electronegativity. So as a result, they don't dissolve in water well. So these are the hydrophobic amino acid side chains. For this course, grade 12 Chem, you do not need to know the details of the amino acids. Okay. I don't think you need to memorize the 20, even for grade 12 biology. I don't think Mr. Mix will make you do that, well, of course, unless I'm wrong. But however, at university, if you decide to pursue this field and you take a course called biochemistry, well, in that course, you're going to have to memorize all 20 amino acids. I'm talking about you got to know the name, you got to know the 3-letter abbreviation, you got to know the 1-letter abbreviation, you have to know the structure of all 20 amino acids. So yeah, good luck if you decide to pursue that. Okay, so a polypeptide will be a long chain of amino acids. Okay, a peptide, why is it called a peptide? Because the bond that holds together amino acids is called a peptide bond. So in the condensation reaction, water is removed, the C double bond, oxygen will be bonded to the nitrogen, which was referred to as a peptide bond. So if you have a long chain of that, it's a polypeptide. Okay, so this will eventually extend for hundreds or thousands of amino acids. You have this long chain of amino acids, but a protein has to be a 3-dimensional blob. Having a long chain doesn't do anything. A protein relies on its structure. So intermolecular forces between the long chain will start to interact and fold the protein to a very specific shape. So first it will coil up or go into a zigzag, so alpha helix to beta sheet. And then more intermolecular forces will kind of fold that on top of each other into a 3-dimensional blob. What type of intermolecular forces? Well, you can have hydrogen bonding between the long chain. On different parts of the chain can have hydrogen bonding with each other. Or you can have London forces. The hydrophobic parts will interact with the hydrophobic parts. Ionic bonding, because some R groups are charged, as well as even covalent bonds. You can have a sulfide bridge that links them together. So all of these forces together fold your amino acid chain into a 3-dimensional protein. And then you can have 4 of these coming together to finally make that big structure. Okay? Like, for example, alcohol dehydrogenase or hemoglobin. They all have their own unique shapes, and their shapes are determined by the sequence of amino acids. Okay, so that's enough about protein. The last macromolecule is nucleic acid. Okay, nucleic acids are the polymer molecules that make up DNA and RNA. Okay, so these are made up of monomers we call nucleotides. And in figure A on this page, you have a picture of a nucleotide. A nucleotide has three components. The pentagon in the middle, the orange thing, is a 5-carbon sugar. Okay, and that 5-carbon sugar in figure B, you have two types. You can have ribose or deoxyribose. There's a small difference. Ribose has a hydroxyl group, and if you just take a look at that number on carbon 2, it has OH. But deoxyribose doesn't have OH, it just has H, so that's a slight difference. Okay, so DNA stands for deoxyribose nucleic acid, whereas RNA is ribose nucleic acid. So that's your 5-carbon sugar. Attached to the 5-carbon sugar, you have a phosphate group, just like the polyatomic ion PO4 3-, you have that right there. On the other side of your sugar, you have a nitrogenous base. And there are 5 different kinds of nitrogenous bases in figure C. They all have a name and a unique structure. We just refer to them by the first letter of their names for simplicity. So C, U, T, G, and A. U only exists in RNA, T only in DNA, but that's a small detail that you don't need to know for chemistry. So just know that a nucleotide has a phosphate group, a 5-carbon sugar, and a nitrogenous base. So how does this make DNA? Well, they are linked together with phosphodiester linkages. Right? So phospho, meaning phosphorus, diester, meaning you have 2 ester linkages. All right, so DNA is made up of a backbone, and the backbone is composed of alternating phosphate sugar, phosphate sugar, phosphate sugar. So we call that the phosphate sugar backbone. The sugars will all have a nitrogenous base sticking out. Okay, in DNA, it could be one of the 4 letters, A, G, T, C. Okay, and the sequence of these bases spell out a message. Okay, literally, 4 letters in the alphabet, you can spell out different words and information. So that's how DNA codes for information. And we have double helixes for DNA. So you have a complementary strand that basically hydrogen bond with each other to form 2 strands. And more hydrogen bonding will turn it into a helix shape. So that's how you get the structure of DNA. So you need to know that DNA is a double helix. So a helix meaning spiral, double because you have 2 strands, held together via hydrogen bonding between the nitrogenous base pairs. And the backbone is composed of a polymer of phosphate and ribose. Okay, so via phosphodiester linkages. That's the extent you need to know about DNA. Okay, you do not need to know all that detail you learned in grade 12 bio. This is the chemistry version of that. All right, so do you have any questions? Awesome. So this concludes this lesson. In this class, we talked about condensation polymers. So we have synthetic condensation polymers, so polyester, polyamide, and how to identify the monomer and how to draw the polymer of those. And then we looked at natural polymers, the 4 biological macromolecules. We didn't really look at lipid all that much. They're just carbohydrates or polysaccharides, proteins or polypeptides, nucleic acid. So basically polymers of nucleotides. You need to know the linkage that holds them together. Okay, that's pretty much it. So if you don't have any questions, we will end it here.